Hello, I'm Dr. Damon Smith, CEO of TALLC, a new ophthalmic medicines company using our novel anti-inflammatory analgesic drugs delivered by smart cell technology to treat medical conditions throughout the eye. Our lead indication is keratoconus, an orphan disease usually first seen in teenagers that causes a progressive thinning of the cornea. This then leads to weakness and the cornea starts to bulge, distorting vision. If untreated, this can lead to sight loss. There are really only two treatment options for keratoconus: corneal transplants with all the issues of transplanted human tissues, and cross-linking surgery, which chemically strengthens the cornea and can halt progression. Unfortunately, while effective, the surgery is extremely painful, which tends to limit the eligible population. Let's talk more about the importance of pain control, and in doing so, refer to the doctors and patients who know most about it. Here then is a quote from Marie-Claude Robert, a leading Canadian ophthalmologist, which I think speaks for itself. Patients we've contacted are equally clear, and it's obvious that fear of surgery is delaying early treatment. If we could treat the pain effectively, we believe that more people would have the procedure and earlier, and outcomes, both short and long term, would be better. Unfortunately, we can't do this well today, since oral opioids, quite apart from the addiction risk, are ineffective. Topical NSAIDs delay wound healing, increasing infection risk, and steroids don't act fast enough, so they can also delay healing and increase intraocular pressure. We should now talk about TALLC's solution to effective pain control for keratoconus surgery. That is our new drug, which we called TALLC, or A001 for short. A001 stimulates human CB2 receptors, which are found throughout the body and extensively on immune cells, where they have an important role in controlling inflammation. They're also found in the CNS and generate analgesia when activated. A001 binds more tightly to human CB2 receptors than any other receptor found in the body, and we've shown it to be 10 times more effective than NSAIDs in pain control models, where it begins to work very quickly and to produce long-lasting effects from a single administration. In terms of mechanism, as we'll show in the following slides, A001 reduces levels of pro-inflammatory cytokines and adhesion molecules, and it stops immune cells from binding at the injury site. We deliver A001, our new anti-inflammatory analgesic, using our proprietary smart cell technology. These are 30 nanometer micelles, neutral micelles, which allow deep penetration within the cornea. They allow transcorneal penetration, so you can access the aqueous humor and the trabecular network, and to the iris as well, and we believe getting into the vitreous humor. Of course, topical administration to get to the back of the eye is a holy grail, but we believe that A001 delivered by smart cell has the opportunity through topical administration to get to the back of the eye and to treat retinal diseases. It's a proprietary and unique technology, and we have issued patents out to 2038. In this first slide, we show the effect of A001 in reducing pain after surgery in a mouse model of analgesia. You can see the analgesic response at the side and increasing doses of A001. The middle dose, around 3 to 4 milligrams a kilogram, or the EC50, is around 10-fold lower than that of an NSAID we also used in this model. So you can see from the ED50s, much lower ED50, much lower, smaller amounts of drug are needed to achieve analgesia in this model than an NSAID. If we move on to a model of postoperative pain in the eye, a corneal hyperalgesia surgery model, in this model we added A001 to the front of the eye after trauma, and we compared it to the classical CB2 agonist HU308. As you can see here, the lowest dose at around 0.25% is around 12 times more potent than the classic CB2 agonist HU308. So after topical administration, A001 effectively reduces pain score in the eye. Since a picture paints 1,000 words, here is a picture of a slide showing two sections of the cornea and the number of leukocytes or immune cells that bind in the presence or absence of A001. On the left-hand side, we see the number of leukocytes, immune cells, which have accumulated in the eye after trauma. And on the right, we see when A001 or A001 is added. As you can see, strikingly fewer numbers of leukocytes, far less immune cell binding, and enhanced CB2 agonism when we add A001 to that model. Let's talk about the product itself now, the product profile, the product value as perceived by others, and the product development and timelines that we have. We first put together a product profile for TA001, as you can see here in the bullet points. First of all, to stress that it has a unique novel mechanism of action, different from NSAIDs, different from corticosteroids, and different from the opioids that are currently used today. It's a novel mechanism, CB2 analgesic. It's for topical installation. We believe it will be given the day before surgery and then 3 days afterwards. And as we've shown, it has superior potency to NSAIDs, but key has no psychedelic or narcotic effects. We believe these anti-inflammatory responses induced by A001 can result in reduced scarring and corneal haze. And we've already shown that a single administration can give rapid and sustained effects. Most importantly, since it's a different mechanism of action from NSAIDs and corticosteroids, it has no effect on wound healing or on intraocular pressure. That can give improved pain management without those side effects, an improved patient experience, and therefore decreased anxiety and the ability to have earlier intervention for a wider population. Now, we showed this target product profile to a series of US practicing ophthalmologists, 7 to 28 years, up to 400 keratoconus patients seen. And they had a very consistent set of responses. They spoke about the problems that they're experiencing today, the unmet need. As you see on the left here, topical NSAIDs inhibit healing and I don't get pain control. They spoke about the ability to widen the population, the number of patients who would benefit from keratoconus surgery if they had this product available. We would offer it to more people. This would allow us to do more procedures. The improved patient experience, which is very important, and a reduced need for narcotics. But also very clear was that they felt that the product profile, the value of A001 was far greater than just keratoconus. Indeed, this ophthalmologist at the top mentioned this could reduce the risk of dry eye following the surgery. And that's a general point that we're seeing coming out. 94% were extremely interested in using the product, but all of them, in fact, the 6% who said they were only moderately interested said that because they felt the indication was too narrow. So if you look at the statements in the box at the bottom here, of course, they would use it for their cross-linking patients, CXL standing for cross-linking, the chemical cross-linking. They would use it for all 10 of their next patients, but they stated it's a game changer for practices performing corneal surgeries, i.e. not just keratoconus surgeries, but surgeries outside that indication. So let's look at the wider opportunity. We believe if we can treat the unmet need of pain in keratoconus, we can access far more patients and earlier. We believe that's around a $0.8 billion opportunity for this product in North America. Looking to the wider population and the wider number of surgeries, the PRK and LASIK and cataract surgeries, well, that's around 8 million eyes, and we believe around a $2 billion opportunity for the product. We also believe that A001 will have value in chronic diseases. That's glaucoma, dry eye, as mentioned before, macular degeneration, and diabetic retinopathy, which represents around 60 million American eyes and an opportunity of around $3.1 to $3.5 billion. That's for A001 and our pipeline drug candidate TA002, which is a CB1, CB2 receptor agonist. Why do we believe we'll have value at the back of the eye? Well, CB2 receptors are found throughout the eye, both in the cornea, as we've explained, but also, as you see here, in the uvea, in the trabecular meshwork, controlling intraocular pressure, perhaps, and in the retina, where inflammation is shown to be an issue with AMD and diabetic retinopathy. The ability to bind to CB2 receptors there and reduce inflammation, we believe, can have significant value for treatment of those diseases, perhaps without the need for intraocular injections. In terms of where we are, well, we're a preclinical company. We are seeking investment to help us get from where we are today through IND-enabling studies and into phase 1 human trials, where we'll look eventually into phase 2 for keratoconus surgery and for PRK. We also will use those funds to help further the development of A001 and the follow-on molecule A002 in retinal disease. In parallel, we're always looking for co-development and licensing partnerships with specialty ophthalmology companies, again, to help the speedy and directed development of this new molecule and new delivery system in the treatment of ocular diseases. I'd like to thank you for listening to our presentation here, and if you would like more information about TALC, please look at our website or contact us on the link below. Thank you very much.